Let's start from the preparation of Arabidopsis plants. Those are 1 week old, 2 weeks old, and 3 week old plants grow on Jiffy. And you can have an overview of those plants grow in a tray. And this is a 4 week old plant. You can grow them in defined medium like Jiffy or soil mix like Metro Mix 360. Those are 4 weeks old Columbia white type plants. You can also grow them in Fafa. Those are also 4 week old Columbia white type plants. They all look healthy. We make sure we have healthy plants. We can start the next step of preparing enzyme solution. This is cellulase powder I add into a preheated mannitol solution. And I use a plastic loop to try to dissolve the enzyme solution quickly by stirring it. Usually within a minute that you can dissolve most of the enzyme powder, and you can see that you get a clear enzyme solution. We have cellulase R10 and mesolyzine R10. And then we heat the cellulase solution at 55 degrees for 10 minutes. The purpose is to inactivate DNase and proteinase in the solution. After heating, then we add the tube into ice bucket to lower the temperature to room temperature, and then afterwards we bring it up in a syringe. Then the next step is to use a 0.45 micron filter to clean this enzyme solution. So this is the 0.45 micron filter we use, usually available in the lab. And the purpose is to get a feeling that the enzyme solution is dissolved completely. Then you can push the solution through the filter very smoothly without much resistance. And another thing is, if you want to have sterile condition, this is the step that you can make sure the enzyme solution is sterile. However, for our protoplast transient assays, most of the experiments is done within 3 to 24 hours. Under that condition, we don't really need to keep everything sterile. Now we'll start cutting the leaves. Those are cotyledons, and the first, second leaves, the third and fourth leaves, then the fifth, sixth, the seventh leaves. Usually it's the fifth, the sixth, and the seventh leaves in this typical 4 weeks old Columbia plant. It's what we use for protoplast preparation. I cut the leaves at the end of the petioles, put it on a piece of paper, and use a one-side razor to remove the top of the leaves. We usually use the middle part of the leaves. And I cut it to between 0.5 to 1 millimeter leaf strips. We only use fresh leaves, never reuse the razor blade. Okay, we also don't use the bottom of the leaves, basic to keep a homogeneous population of leaves. Strips, only the middle part of the leaves. We take the leaf strips, you dip on one side first and flip it, and then dip it on the other side and release the leaf strips into the solution. This is a very important step. Basically, you need to have the cutting edge immediately exposed to the solution before it seals. Now again, dip one side and flip, and another side, and then allow the leaf strips to be separated in the solution. And then if the leaf strips stuck together, you can use plastic loop to separate them gently. Try not to wound the leaves during the process. So this is the beginning of leaf digestion. Then we usually take the plates into a desiccator to apply vacuum infiltration for about 30 minutes. This is to help in infiltration of the enzyme solution. PEG solution will be prepared now, right after the beginning of leaf digestion. We add the PEG solution into mannitol and calcium solution. It will take a while to dissolve PEG, so we'll prepare PEG and give it enough time to dissolve completely. The PEG we use is specific. It's Fruca PEG 4000. After 3 hours, you can see the leaf strips have been digested. They become not as stiff, and the best way to test to see whether they're well digested is to take a few pieces and smear them on the top of the paper. If you can smear the leaf strips easily, that means they're well digested. And then we will start the process of releasing the protoplast by gentle shaking and swirling. Okay, and then you can see the solution turn green. The next 40 seconds will take you to watch the release of protoplasts from the digested leaves under microscope. Now we're ready to harvest the protoplasts. Usually we add some W5 solution, which contains a lot of calcium. Arabidopsis protoplasts, they're happy in calcium solution, and having more calcium in the medium, it's also easier to centrifuge the protoplasts. So we add W5 and mix, and you can see the green protoplast in a solution. We mix it well before we use a nylon mesh. Usually it's the 75 micron nylon mesh we use to filter the protoplast through. So this is the 75 micron nylon mesh. Then we pipette the protoplasts through the nylon mesh gently. You don't really need to cut off the tip of pipettes. For those protoplasts, they're pretty tough that they will not be break even if you use regular pipettes. Just do it gently. Don't pipette up and down too quickly and too harshly. Okay, so we take the filter out, you clean it, and then we prepare a balance tube for centrifugation. For protoplast centrifugation, we use as slow the speed as possible. In this case, we use the all clinical centrifuge on the bench top. Typically, we use at speed 3 to 4 for about a minute or two. And then after the centrifugation, you can see the green pellet at the bottom. This looks like a very nice protoplast pellet to me. The solution is still a little bit green, mainly because there are some broken cells during the preparation. We will remove the supernatant as much as possible by pipetting. And the green pellet should resuspend easily by gentle shaking. almost instantaneously. If your pellet doesn't resuspend immediately, that means they're broken cells, so you don't really have good protoplasts. Next, we will do protoplast counting. This is a very important step. Before DNA PEG transfection, we need to know how many protoplasts we have obtained. We pipette 9 microliter of protoplast solution underneath the cover glass into the monolayer chamber and use the regular microscope to count protoplasts using the hemocytometer. This is a hemocytometer using object 10, 20, 40. And what we would like to do is to look at the protoplasts with the help of hemocytometer. We count the protoplasts in the 25 squares. Each square is 200 micron in length. This is the large square. In each square, there are 16 small squares. Each has the length of 50 micron. And Arabidopsis protoplasts, mainly mesophyll protoplasts isolated from leaves, they are various in size. Usually they're about 25 to 50 micron. So this size of hemocytometer is perfect to estimate the size of Arabidopsis protoplasts. So those are just representative size of Arabidopsis mesophyll protoplasts we can see using hemocytometer. And we count protoplasts that located within this 25 large squares. To count the protoplasts we obtain in the solution, assuming we obtain 100 protoplasts, then we times 10 to the 5th per ml. That gives us 1,000,000 cells per ml for the starting solution we have. In this case, we actually have only 0.5 ml of the protoplast solution to start. So therefore, our yield is 5 times 10 to the 5th total protoplasts. And this is a preparation from 10 to 12 leaves that we cut at the beginning. And then we want to make a final solution of 2 times 10 to the 5th per ml for protoplast transfection. So then we'll resuspend this originally dense protoplast solution to final 2 times 10 to the 5th per ml. That's our starting protoplast solution concentration. Okay, and protoplasts should be resuspended easily. These are 6 well, 12 well, and 24 well plates. This is 5% calf serine. We can choose to coat the plates with 5% calf serine. We pipette 1 ml 5% calf serine into each well. The purpose is to prevent protoplast sticking to the bottom of the plates. We shake the plate to make sure the liquid covers the surface of each well. Then we pipette it up. It's very simple. We usually prepare the culture plates before we start the protoplast transfection with DNA and PEG. This coating procedure is optional, not absolutely necessary. Then we add 1 ml mannitol solution for protoplast culture later. You can also use W5 solution if you like. Depends on your experimental purposes. Okay, now plates are ready. Then we can start DNA PEG transfection with protoplasts. This protoplast been resting for about half an hour. You can see the cells are well pelleted at the bottom. Then we'll take out the supernatant. Okay, first we suspend the protoplasts. Then we add magnesium mannitol solution at room temperature to resuspend protoplasts before DNA transfection. The use of magnesium mannitol solution enhances the transfection efficiency. then when we start, we will pipette the DNA into tubes. I will not be too greedy at the beginning. Try to work with maybe 6 samples at once. Okay. After DNA pipetting, then you can start pipetting protoplasts. Each time during protoplast pipetting, you need to resuspend protoplasts. This is at 2 times 10 to the 5th per ml. You add protoplast into the tubes, mix it with DNA gently, then we pipette PEG calcium solution. It takes a little bit longer to pipette PEG solution because it's viscous. Then you can see there are 2 layers of solution, protoplast green part on the top and the PEG at the bottom. Then you mix it gently and let it transfect for at least 5 minutes. It can be as long as 30 minutes. Then you can add W5 solution, in this case 400 microliter, into the transfected cells to stop the reaction. And all you have to do is just invert the tubes gently. Okay, now it's ready for centrifugation. Again, we use benchtop clinical centrifuge. Again, we centrifuge for as low the speed as possible, usually speed 3 to 4 for a minute or 2. Then you can take the tubes out. Then you can see clearly the green nice pellet at the bottom for your transfected protoplasts. That means the transfection is successful. If the cells are not good, you don't see a nice pellet. Then we will suspend the solution, the protoplast in culture solution, either in mannitol or W5. Then transfer the solution into culture plates. Each plate, as I mentioned, contain 1 ml culture medium. This is the condition where protoplast can breathe easily and stay alive. The tubes we use are round bottom 2 ml tubes. Okay, so to allow you to learn this procedure, we're going to repeat this procedure again. Starting with DNA pipetting. In every tube, we add 10 microliter of DNA solution, and that's for every 100 microliter of protoplast solution you can add. Again, before pipetting protoplast, we always have to resuspend the protoplast because they're heavy and they can settle to the bottom of the tube easily. Now you can see the protoplast much more clearly because this is 5 times more protoplast in the solution than what we used in the previous experiment. This is mainly just to allow you to see the procedure more clearly with more protoplast as the solution is greener. We add protoplast to DNA, mix it, and now we add PEG solution. So you can see the PEG solution sink to the bottom. There are 2 layers, and then you can mix it gently. That's all you have to do. Again, we're suspending protoplasts before pipetting. Add protoplast to DNA solution, mix it. Very simple. Then pipetting 100 microliter of PEG calcium solution. This pipetting takes a little bit longer because the PEG solution is viscous. So you can see the PEG solution sink to the bottom. There are 2 layers, protoplast and PEG. Mix it well until you don't see the separation of the 2 layers anymore. Just a few seconds. Then we add W5 after the transfection to complete the procedure. After after adding W5, you just simply invert the tubes to mix everything gently. Okay, that's done. Then we centrifuge the tubes again at speed 3 to 4 for a minute or two. Now you can see more protoplasts. As I say, we use 5 times more protoplasts for this transfection procedure for better visualization. You take out the supernatant that you don't want that contain PEG, and you resuspend the pellet gently. Okay, very simple procedure. This is another sample after centrifugation. Take out the supernatant carefully. Leave very little in the tube. Try to be consistent among different tubes. Resuspend the pellet gently. It should resuspend very easily. Now we add between 50 to 100 microliter of W5 or mannitol solution for resuspension the protoplasts before transfer them into the culture medium for incubation. Okay, you can use regular pipette tips for pipetting protoplasts for all the procedures and mix them well. Then for different purposes, you simply wait for 3 to 8 hours. Then you can start harvesting the protoplast for your next experiment. That's it. Very simple.